Hello and welcome to Lab to Launch, the seminar series for researchers interested in pursuing entrepreneurship. I'm Sarah Jane Murdoch, Administrative Project Coordinator for the Institute for Innovation and Entrepreneurship at UT Dallas and Operations Manager for the Venture Mentoring Service of North Texas. As we move through today's session, I invite you to post questions into the Q&A box for our guest speakers to answer. Our host is Bryan Chambers, Vice President, Accelerator and Investment Fund for the Capital Factory and Director of the UT Dallas Seed Fund. Our guest speaker for today's session is Dr. Eric Olson. He is the founding chair of the Department of Molecular Biology at the University of Texas Southwestern Medical Center and the Center for Regenerative Science and Medicine at UT Southwestern. Dr. Olson's discoveries at the interface of developmental biology and medicine have illuminated the fundamental principles of organ formation and have provided new concepts in the quest for cardiovascular therapeutics. And now here is Bryan Chambers and Dr. Olson.
All right, thanks so much, SJ, and welcome everybody. As you heard, for those who might be tuning in for the very first time, this is our Lab to Launch series where we visit with some of the top scientists and inventors in the North Texas region to hear about their journeys as entrepreneurs commercializing their novel technologies. And I must admit, this is my first time to throw down with a molecular biologist. I'm super excited about it. The only thing I wish is that I had a few more weeks to prepare because I am truly fascinated by the work of our distinguished guest today. In 1995, Dr. Olson founded the Department of Molecular Biology at UT Southwestern Medical Center in Dallas, where he's a professor and the chair of the Department of Molecular Biology. He also holds the Robert A. Welch Distinguished Chair in Science, the Annie and Willie Nelson Professorship in Stem Cell Research, and the Pogue Distinguished Chair of Research on Cardiac Birth Defects. He's dedicated his whole career to deciphering the mechanisms that control the development and disease of heart, cardiovascular systems, and the skeletal muscle tissue. Two fun facts we now know about Eric from our research are that he's one of the most cited researchers of all time, with his publications cited over 90,000 times throughout literature. And he's trained numerous students and postdoctoral fellows, many of whom are emerging as the next generation of leaders in cardiovascular biology. And also in his spare time, he plays both the guitar and the harmonica with the Trans Activators, a rock band inspired by Texas icon Willie Nelson, who created the professorship that Dr. Olson holds today. So those are just a few of the reasons why I'm super excited about today's session with Dr. Olson. Dr. Olson, thanks so much for being here. I gotta ask, where did you grow up, and throughout your background, were you always into science?
Well, first of all, thank you so much for having me today. I really look forward to this conversation. I grew up primarily in North Carolina, although I moved around a lot as a child with my family. And as you ask, I was always interested in science from an early age. I remember in elementary school, we'd go to the library and you could choose which books you wanted to read. And I loved to read books about space exploration and aspects of science. So I always knew from a young age that I would go into a career in science, but I never knew what form that would take until much later in life.
So I also read that you grew up in an incredibly musical family. Of course, that makes a ton of sense why you play both the guitar and the harmonica. But as you reflect back on your life and your career, was there anything in particular that was the driving force that encouraged you to pursue the field in particular of molecular biology?
Well, I grew up in a musical family, as you said. My mother and her mother were professors of music at the Eastman School, so I grew up playing all sorts of instruments. And my father was a chemist, and he really instilled in me a love for science. And I think it was the two of them that both influenced my future interests and the direction that I would take.
Yeah, so let's fast forward. At UT Southwestern, really you and your colleagues went on to discover many of the key transcription factors and the mechanisms responsible for cardiac gene regulation and formation of the heart and other muscles. And your work also provided the genetic blueprint for tissue formation that unveiled the molecular underpinnings of the congenital and acquired diseases of the heart. you also discovered the epigenetic mechanisms and microRNAs as regulators of muscle development and disease. So first and foremost, let's take one step back. Can you tell us the story? How and when did Willie Nelson get involved with your lab? And the question that I've got to ask is, was it your love for music, or was it his passion for molecular biology that brought you two together?
Those are interesting questions. I got to say, it's humbling to realize that most people are more interested in my role with Willie Nelson than in my science, but I get that. Willie was always a musical and personal hero of mine, and I never met him, never thought I would meet him actually. And then one day his wife Annie, who I have come to know quite well, was bringing their children to UT Southwestern for an exam, a pediatric exam. And the president of the university, knowing that I had a great interest in music and was playing in a band, asked me if I'd like to meet with them. And so I met with them and talked to them about what we were working on and really hit it off. And then Willie decided to have a benefit concert and raised some money. It created an endowment that supports some of the work in my laboratory. And over the years, I've had the opportunity to spend quite a bit of time with Willie and his wife Annie on the tour bus, before concerts, or at their ranch outside of Austin and elsewhere. So it's been really a very exciting experience for me to actually meet and spend time with one of my all-time musical heroes, and to hold the Annie and Willie Nelson Chair in Stem Cell Research, which they funded, is really something that means a lot to me.
Well, what an awesome and unlikely partnership that's really gone on to probably impact your research, the department, the school, but also many of the lives of those that Willie and Annie had touched as well. I've heard about this stem cell guitar. What's the story, or what's the background on that? Is that just a guitar that is that a special guitar from a special experience? What's the stem cell guitar?
The stem cell guitar it's a Martin guitar, which is a very good acoustic guitar, similar to the ones that Willie plays. And I was down at their ranch, and Willie and Annie autographed the guitar and took a lot of pictures, and it was a great experience there. And then so we hung up the guitar in the hallway outside my office. It's been a great source of interest whenever any politicians, donors, friends, colleagues come through. They always like to look at the Willie Nelson guitar. It's kind of become the touchstone, or the symbol of our department and our stem cell center. So it's having that guitar is just one of many fascinating experiences that I've had with Willie and Annie.
That's so cool. So Dr. Olson, you started your first company, Myogen, in 1999. Do you remember there being a defining moment when you knew you were ready to, commercialize the first research and start your first company out of UT Southwestern? Can you tell us a little bit about what Myogen was and how and when you kind of knew you were ready to do this your first time?
Yeah. So my lab, as you have pointed out, has worked on the basic biology of muscle and the heart. The students and trainees in my lab discovered many of the key regulatory proteins and genes that control the formation of muscle tissues, and along the way it became apparent that many of the genes and mechanisms we were working on were not only important for development, but also were important in various diseases of muscle, in particular heart diseases, which, as you undoubtedly know, is the #1 cause of death and disability in the industrialized world. And I've always been interested in pushing our work beyond the limits of knowledge and seeing how far we could extend it. And it just seemed to me at that time that there were opportunities to try to move our work in a direction that had more clinical application. So I was fortunate to be able to team up with two colleagues of mine who happen to live in Boulder, Colorado, and we formed a company called Myogen. My lab had discovered a gene regulatory protein which we named myogenin several years earlier, and it was a master gene for muscle, and so Myogen was a derivative of that name. And we started that company, and I was so exhilarated by this new experience, just to meet new people, to learn new things, to see how far our basic science work could go. I never lost and never have lost my passion for my academic laboratory and the basic work that's been done here. That's really the source of my energy and passion. But this was an opportunity to add a new dimension to it. And so several of the discoveries in my lab served as the basis for the launching of Myogen. Several of the really great people from my laboratory went to that company as well and helped to set it up. And along the way, we in-licensed some other drugs, and it was a really interesting experience. We had a lot of ups and downs, learned a lot. I remember that the state of Colorado was trying to attract biotech, and they had an abandoned military base there outside of Denver that they were trying to turn into a biotech park. And so they offered us a little bit of space in there. We had just a couple of rooms with folding chairs and a coffee table, and that was all we had. And we just started trying to raise money, and it was very interesting. I remember going to Wall Street, where I'd never been to Wall Street, and I remember standing in a big conference room at the top of one of these buildings on Wall Street, talking to a whole room full of investors and bankers, telling them what I wanted to do. And I remember one of them said, "This is nothing but a bunch of molecular gymnastics." And so I'm like, okay, does that mean you don't want to fund me? But anyway, we ultimately got funded, and we raised money, and we actually went public, and had some ups and downs. Then we had some real big ups, and ultimately the company was acquired in a $2.5 billion buyout by Gilead Therapeutics. So that was my first entree into biotech. I really learned a lot, although I had much more to learn after that.
Yeah, that's awesome. Well, first one, it ended awesome. Pretty rare to happen. That your first one probably goes like that. But tell us about what it was like in the late 90s to do a deal and commercialize tech and research out of UT Southwestern, and maybe to leapfrog, like, how has that changed today? Can you talk about that a little bit? What was it like then, and what role, other than your colleagues and some of your students getting involved, was there anything specific that UT Southwestern did to support your new business and commercializing it? And what's that like today?
Yeah, there was no biotech to speak of in Dallas at that time. And while there were academic investigators here in Dallas that were involved in pharmaceutical company consulting and other things, they weren't necessarily involved in founding companies from scratch. And I've always been kind of a pioneer. I like to discover new things and push beyond the boundaries of knowledge, and I tend to have an independent streak. So I really was attracted to the idea of starting companies rather than joining pre-existing companies. At UT Southwestern, they enabled me at that time to do what I wanted to do, and I was given advice if I needed it, but pretty much they left me to do my thing, and it worked out really well at that time. And as you pointed out, I was able to start 3 other companies subsequently. And over those years, that's been about 20 years now since we founded Myogen. Over those years, Southwestern has really become very, I think, entrepreneurial. I think there's still room to grow in that area for those people that are interested. But there is a lot more interest in this area, and our technology transfer office here, I think, is first rate. It's being led by Claire Aldrich, who's done just a spectacular job with that. And there is a lot of interest, I think, here for people to start companies. Now, everybody doesn't need to start a company, and every type of science doesn't lend itself to translational development. Basic science is just as important as translational science. But I would say that from when I started Myogen up until today, there was not a culture of entrepreneurship at UT Southwestern as compared to the coasts. For example, if you're at Harvard or UCSF or Berkeley, every other faculty member is involved in a company, and there's just a whole different entrepreneurial spirit there, and there's a lot of biotech companies there. And here that hasn't yet been the case, although I think it's gaining momentum, and I'm really bullish on trying to attract biotech here, and I think that would enhance the academic enterprise as well by providing an outlet for discoveries and also enabling the recruitment of a lot more scientists to Dallas.
Well, thank you. Yeah, we'll talk about the changing ecosystem. I've got some questions about that in just a moment, but could you share with us one thing? What is it that you wish you knew your first time, like that you know today, when you were starting out your very first company? What is it that you wish you knew if you were doing it all over again? If you could tell yourself back in, in the late 90s, is there anything in particular?
Yeah, well, I guess there's a scientific and a financial dimension that I could mention that might be interesting to some people. The scientific dimension, and that is, while most or all science is interesting, most or all science isn't applicable to building a company. So there's lots of really interesting things, and that people can work on. This is one of the key differences between academia and biotech. In academia, anything's interesting, can be interesting if you dig into it and it's something new, and it doesn't have to have a commercial endpoint. But in biotech, it's really got to be focused and directed toward a goal and working on a time clock, and you've got to have a path forward to a product. So I think I've learned that along the way. I didn't necessarily know that in the beginning. And I would say the other dimension, to answer your question, might be the financial dimension. And that is, it's very important, especially for inexperienced academic investigators, to make good financial decisions when they're starting a company, or to get good advice from people who have that experience, because the investment community, they can be very savvy. They know a lot about finances, and scientists don't necessarily know that. And I think it's important to team up with very strong investors who share a common vision with the founder of the company, and who really are in it for the long haul and in it for the right reasons, not necessarily in it to flip it and make a quick buck, because it's very tough to develop drugs. It's unpredictable, and you don't necessarily know how long the timeline is going to be, and it's really important to have a strong commitment from the investment team.
Yeah, thanks. Thanks for sharing the context. And so Dr. Olson, you went on to do this several more times with Myogen, I think is how you pronounce Myogen Therapeutics.
Yeah.
And then even most recently, again in 2015, with Exonic Therapeutics, which was acquired for $1 billion. But I noticed that with Exonic Therapeutics, you also relocated this business, if I'm not mistaken, to Boston, as you just mentioned, to the Northeast coast. Does Dallas or North Texas have what it takes to build and create great biotech companies here?
I think Dallas has what it takes. It's first of all, I love Dallas and I love Texas, and I'm not ever leaving, even though people would like me to move to the coasts. But I am really attracted by the entrepreneurial spirit, the can-do attitude, the positivity of Dallas and Texas. And those are essential ingredients for starting any company. Dallas has always attracted visionaries, and we all know many visionaries in Dallas who have started great companies and done great things in many industries. Unfortunately, so far, biotech has been relatively limited here, although there are certainly some success stories. But I think we have the ingredients. There's a lot of investment, finance, and capital here in Dallas, and we've got a world-class academic medical center, which is an essential component to any biotech community. So we have the ingredients, but it's going to take some further effort to get a few more success stories and to just build the ecosystem. You're correct that with Exonics, that was the fourth company that I had started, and I did it a little different. Every one of them was a little different, but that one, I decided to do it by myself to see whether I could start one by myself, and I teamed up with a venture philanthropy group called Cure Duchenne Ventures, which is a venture philanthropy fund that supports startup ideas in Duchenne muscular dystrophy, which is a muscle disease that's debilitating and it causes boys to end up in wheelchairs and on respirators, and that was really the focal point. And one of the points I think I should make about starting a company is that you really want to have a lot of confidence that the idea you're pursuing has a good chance of working. Now, you can never be sure, but you really want to be as confident as possible. And in starting Exonics, what I wanted to do was try to cure this disease by correcting the errors in the DNA that cause Duchenne muscular dystrophy, DMD, using CRISPR, which is a gene editing tool that can modify the genome, and which was recognized by the awarding of the Nobel Prize this week to Jennifer Doudna and Emmanuel Charpentier. So I felt like this could really work based on some groundbreaking work by trainees in my lab, but I wanted to be even more sure, and to do that I wanted to see whether we could correct this disease in a large animal, and that is in a dog, a breed that has the exact same disease as humans. And that was going to take some money to do it. It was beyond what we had in my laboratory. So the patient advocacy group provided the funding for us to do the experiments to see if it could work in dogs, and lo and behold, it did work in dogs. And armed with that information and the startup funds from the patient advocacy group, we were able to launch the company and attract major funding from the Column Group, which is a venture fund out of San Francisco. Now, you mentioned that I put the company in Boston instead of Dallas, and that was it was a decision that I wrestled with a lot, and that wasn't always a popular decision. But in this particular disease space, it's very competitive, and I felt like we didn't have time to waste. And in Boston, there was a lot of expertise there that we could use to build the company, whereas in Dallas, it would have required recruiting all these people to Dallas, and that could take a lot of time. So we put it in Boston.
So some people may know, others may not. Earlier this year, a new initiative called Pegasus Park was announced, in partnership, I think, with lots of different groups, but principally UT Southwestern, Lyda Hill Capital, Lyda Hill Philanthropies. A lot of folks we jointly know are involved with that. How do you think that if you could look out 5-10 years, do you think that's the type of impact and the types of initiatives that will really catalyze and create the change so that people like you build more companies here? What do you think is kind of the opportunity here with Pegasus Park?
First of all, I am very excited about Pegasus Park. Been over there and toured the facility, and I'm very familiar with the plans for Pegasus Park. The Biolabs, which is a startup organization that serves as an incubator for new companies, is going to be moving into the Pegasus Park. The Biolabs was originally in Boston, and actually they provided the incubator for Exonics when we started Exonics in Boston, and I was really pleased when they decided to set up shop down here. I think it's going to be transformative, the Pegasus Park, and I think it's also important because it will bring together many different companies and create a critical mass in a singular location rather than having them scattered all across a large city where people don't interact. And I think the linkage between the Pegasus Park and UT Southwestern is also going to really strengthen biotech in this community. So I'm all in on the Pegasus Park and eager to help in any way I can.
Oh, that's exciting to hear. I know the community's buzzing about it, and a lot of people outside the North Texas region are talking about it too. So we're hopeful that is certainly the case, and I think we're all here to support it. So thank you for that. Dr. Olson, I've got a couple more questions. I'd love to fly through them. We can open this up to the audience. I think there's some questions waiting in the queue for us as well. Can you tell us real quick about, you've started so many companies that have gone on to raise money and have successful exits. At what point should an inventor, a faculty member, really be ready to engage the private investment community for venture money? Is there advice or a particular kind of defining moment that you think, oh, it really makes sense if and when these things are achieved or this type of validation occurs? When should somebody really go out and talk to private investors?
Well, I think the first question that an academic investigator needs to ask him or herself is this something you're really interested in doing? I mean, some people are really attracted to the idea of starting a company, and for some people it's not so attractive. And you've really got to be all in 110%, and you have to be willing to put yourself out there and take a risk and not be afraid of failure, because most times it doesn't work out so well. So if you check those boxes and all of those, you fit all those criteria. And then another important point is an academic investigator needs to be at a stage in their career where they're prepared to take on something like this with the time and commitment that it requires, because academic investigators' first priority should be their laboratory, getting promoted, publishing important work, and training their students and postdoctoral fellows. But if beyond that they have the interest, then I think this is something that's really exciting to explore. But the other dimension that I've already touched on is you have to have something that really you think can go all the way towards setting up a company. It's not just about doing cool science. There has to be a path to an end goal with a timeline to get there.
Yeah, and you've already touched on too your venture or financing partners as among the most critical to sharing the vision and that end outcome. Pretty hard to do though when you're in the most initial stages is planning and kind of putting forth a path that actually goes according to plan. How important are those venture partners, and what should other faculty or other inventors know about speaking with or visiting with kind of private investment firms or VC firms?
I can't overemphasize how important it is to team up with the appropriate venture partner. It's absolutely critical to the long-term success of startup biotech. In selecting a venture partner and who to fund, whether it's a venture capital group or otherwise. It's very important that they understand the technology that they're investing in, that they understand the risks and how long it might take, and that they're fully committed to the long term. And part of the reason that it's so important that the investors have a strong reputation and a deep commitment is because typically you're not going to make it to the finish line with a single investor, and it's going to usually require multiple rounds of investments. And the subsequent investors will be attracted to join the enterprise if they have confidence and respect for the prior investors. But if they don't really know the prior investors, or if their visions and goals are not aligned, it becomes very difficult to raise the subsequent rounds of funding that are often needed to take a company all the way to the end. And so many investors, particularly if they're not educated in the space, may think that this is an opportunity for a short-term investment and a quick exit and making some money. And biotech is very unpredictable, and it can be a really long haul. So it's very important to select the right investors.
Yeah, I think that's true regardless of what type of company you're building, but maybe even more so in biotech. Dr. Olson, so you've done this 4 times. Did you always play the same role as you reflect back? Or did you always play the same role, or did it wildly differ from company to company?
It's differed. Every company's different. The first one, I wasn't very experienced, so I led the science, but I didn't really know much about the business, so I was learning a lot along the way. Subsequent companies, I got more involved, and then the last one, Exonics, which will probably be the last one I ever do by myself. That's the one I said I did it pretty much by myself. I thought I knew how to do everything, and I just wanted to really try it. So in that one, I was negotiating a lot of things, and so everyone is a little different, and the outcome of all of them are a little different, and you can never quite predict how it's going to end up.
Yeah, that's right. Well, I think we've got a couple of questions in the queue. I've got a couple for you off the cuff while we're getting those organized. And I want to go back and double click on something that you said earlier about the Nobel Prize winner, which was awarded this week, maybe last week. But this year's Nobel Prize winner in chemistry awarded actually 2 female scientists for the development of CRISPR as a gene editing tool. In an article, I think it came out last week, you called this a revolution. Can you explain exactly what you meant by that?
Yeah, this is there have been some really defining landmarks in science, I would say, over the past 50 years. One is obvious. One is the sequencing of the human genome and the sequencing of virtually all genomes, which can now be done rapidly. And so that provides the blueprint for all of life. It's all right there. 3 billion letters in the human genome encode everything. Obviously, that has laid the foundation for scientific advancement, biotechnology, drug development, etc. But I think that CRISPR gene editing, which is the technology that now allows us to modify genomes, to correct errors in genomes, to improve genomes, you name it, the sky is the limit. I think that is an equally defining, monumental contribution. And I don't think we can predict where this is going to end up. I think the only thing we can predict is that we don't know where it's going to end up. Certainly it'll be used for the betterment of mankind, I hope. Mm-hmm. But this is just the ability of mankind to change the future course of mankind by altering the book of life is something that I certainly could never have imagined.
It really is mind-boggling to think about it. I could ask you questions all day long. You said it's unpredictable, but I'm going to ask you to make a prediction. So when you think about CRISPR, you think about gene editing. What do you think, maybe if you think 5, 10, 20 years out, what do you think is maybe one or two of the primary ways that you think gene editing will go on to benefit humanity? Maybe beyond what we think is obvious.
Well, I mean, I think what I'm going to tell you is probably obvious. I think it's going to be, it already is being applied to plant biology, so it can be used to improve crop production, prevent plant disease, and of course it's going to be used to correct errors in DNA that cause many devastating diseases. This has already been done now for sickle cell anemia and beta thalassemia. Those are two blood disorders, and they can be corrected because we know the mutations that cause those diseases. And the power of that is the combination of CRISPR with stem cell biology. So in the case of blood diseases, one can modify the gene outside the body in a stem cell, and then can reconstitute the blood system by putting it back into a person. That's called ex vivo gene editing. It's also now being applied to diseases of the eye, blindness and other diseases, in which you can introduce gene editing components called CRISPR into the eye, which is an encapsulated structure, and can correct those mutations. Beyond that, my own laboratory, as I mentioned, we're using what we call systemic CRISPR. That is where we deliver CRISPR to all the affected tissues in the body, particularly in our case, we're working on muscle and heart, using a virus to deliver the CRISPR and correct that disease. But these are just two of the earliest stage disorders that are being tested, and there'll be many, many more. There's also a lot of work, interestingly, going on now to use CRISPR for all sorts of things related to COVID-19 diagnostics and COVID-19 vaccines, and testing people for susceptibility to adverse outcomes in COVID-19. You name it, if there's a biological process that's controlled by DNA, which is all biological processes, then they are amenable to gene editing technologies. So I think the sky's the limit.
That's awesome. Well, I'm going to ask SJ to come up and brief us with some of the questions that are in the queue. We've only got a couple of minutes for them. I'm going to go ahead and steal the first one, though, and just ask you, as you reflect back over your 25 plus years at UT Southwestern, what are you most proud of? What comes to mind?
It's an easy question. I'm most proud of the discoveries of the people in my laboratory. I'm the most proud of the successes of the people that trusted me and my laboratory to enable their careers, and watching their career development is really the most rewarding aspect of my career, without question.
Awesome. SJ, do we have some questions in the queue or what?
Yeah, so the first question is asking, so could you speak to Dr. Olson, could you speak to the projects you are currently working on now? So like maybe a little bit more about your current projects.
We are working on a lot of things right now in my lab. They all focus on developmental biology of skeletal muscles, which are the largest tissue in your body, and of the heart. Beyond using the latest types of technologies for CRISPR gene editing for muscle diseases, we are continuing to search for and identify new genes that control new and unpredicted aspects of muscle biology and disease, and we've discovered some really exciting ones. I should say that discovering new genes and discovering new biology and new processes that have never been conceived of before, to be the first to see an aspect of nature, a secret of nature that's been hidden for millennia, and to be the first person to witness that gene or that process is very, very exhilarating. And so we're working on many of those things. We're also working on strategies to repair the heart through a process known as cellular reprogramming, where we're trying to turn scar-forming cells into heart muscle cells as a way to replace missing heart cells in disease. And so we're just letting our imaginations lead us forward and keep trying to push on the outer envelope of knowledge for as long as we can. And one thing I feel very passionate about is that as soon as we discover things in our laboratory, we provide them to the world. We send out things every day to all sorts of laboratories, and I think that really perpetuates and drives science forward.
Yeah, we definitely appreciate all that you do and that you and your lab do, for sure. So thank you so much for that. I just want to put that in there. The next question is a little more about Myogen. So could Myogen stay in Dallas since related science was conceived here at UT Southwestern? So this must be a UT Southwestern attendee. How was the biotech funding at that time, and how do you see the prospects of startup funding environment now?
So I guess the first part of the question is Myogen, the first company, could it be in Dallas? Well, my in that company, as I mentioned, I co-founded it with two friends of mine, and they both happen to live in Colorado, so I was outvoted. Plus, I love Colorado. I have a home there, so I like to go to Colorado. But it could have been here, and it just, as things happen, the companies I was involved with didn't end up here. The third company I founded, called Tenaya Therapeutics, that was co-founded with a group of investigators in San Francisco, and so we put that company there. If I found another company, I'll put it in Dallas, but we'll see how that goes.
We appreciate that. I'm sure Brian, of course, would especially appreciate that. So our last question, we have time for about one more, is how much time did you spend on forming and running a company versus the science and R&D?
It takes a lot of time to start a company. So I spent a lot of time on it. I really did, but it was so close. All the companies I've been involved with are so closely integrated with the work that goes on in my laboratory. It's mutually reinforcing. And so the lessons I learned in starting companies, I think, enabled things to work well in my laboratory and vice versa. So it's really been, in my case, a symbiotic relationship.
That's great. And we actually just had one more question sneak in. So I will go ahead and throw that one out there. What advice do you have for aspiring biotech entrepreneurs that are undergraduates currently?
I think if you're an undergraduate and you're an aspiring entrepreneur, I would encourage you to learn about what it takes to start a company. Learn about I learned everything in my own case by experience, because there was nobody teaching this stuff back then. Now I think there are courses you can take, but I would learn about what is a cap table and what is a pre-money valuation and what is a post-money valuation, and what is what are the steps involved that it takes to raise the capital and to move a company forward, and what types of people does it take to do that. A company, it's like a football team. It takes a lot of different positions and expertise to make it happen. There's no one person that can make it happen. And so I think I would encourage them to seek out that sort of knowledge.
I love that. What a perfect plug for my class, the UT Dallas Seed Fund. And we wrap an entrepreneurial experience where we teach an entrepreneurship class by giving a lens through the investor's eyes about what they need to underwrite and how do they evaluate the cap table. And I think it's a great experience for those emerging to really understand what does it take to win that money from an investor, and what do you have to navigate as an investor. So sign up for the class if you are interested in that type of entrepreneurial, those types of entrepreneurial insights. I think we're right at the 12:45 mark. SJ, is it time to jump into our other session?
It is. Thank you so much, Bryan and Dr. Olson, for joining us today. And yes, so stay tuned. Directly after this, there is a link in the chat, UTD link/lab2launch booster session, where you can meet Bryan and meet Dr. Olson directly. Thank you so much for giving us this unique insight into taking your research from the academia to biotech. So thanks again, everyone, and we will see you momentarily at the booster session.